Hello class, we are now on lab number 3 of the course. This one focuses on how genetic mutations, of which you've seen in DNA, get propagated through biological systems through sexual reproduction. We'll be looking at how that process works, and that process is known as meiosis. And essentially, meiosis is the reproduction of sex cells to produce gametes that go into reproduction. And it's in that process that mistakes are created in the DNA. And then those mistakes are randomized and distributed across different gametes. So whatever mutations an individual will have going into meiosis will be as randomly generated across the gametes and randomly recombined later in sexual reproduction to produce as much variation as absolutely possible. So in other words, meiosis is also the heartbeat of what Darwin was always looking for, the source of all variation. Right. So within this, we'll be learning some things about Gregor Mendel. For those of you who know a little bit about him, we'll be exploring some of his earlier works to try to determine, how things are randomly generated. But I will mention this: if there's anything that you want to pay attention to in this lab, it's the law of independent assortment. Write that down: independent assortment. And there's other things like segregation, all the things you're going to be looking at, but it's independent assortment. That's the one that really determines how things are randomized throughout the entire system. All right, so write that down and make sure you know that, for testing purposes. Okay. Now, additionally, we're talking about the inheritance of acquired characteristics, or of how we're going to get mutations. We're going to be looking at frequencies. What are the odds of getting something? How can we actually predict how genes are going to be moving through a system? And we'll be looking at through the lens of a particular disorder called sickle cell anemia, and looking at how we can predict where sickle cell anemia will strike or not. And another thing we're looking at is trying to figure out, when we talk about is something a diseased condition or not. We're not quite sure with sickle cell anemia. I think once you look at this and the material I have, you'll be questioning the idea of what an actual disease is, right? Disease may actually depend on complete and total context. And the word behind this, and the key term behind this, is called the heterozygous advantage. Okay, so we have law of independent assortment, and we have the heterozygous advantage. And the heterozygous advantage might mean that our system, which is based on dominance and recessivity in our genes, may have some benefit, some protective advantage for populations. And we have to remember this too, that whenever we're talking about evolution, we're not talking about benefits to individuals. We're talking about benefits to populations, right? I mean, bad things can happen in inheritance. We can get genetic disorders and diseases, but overall, what is the effect of even having negative alleles or genes within you that can recombine to produce other things that can actually protect populations? That's what we want to keep an eye on in this lab. Okay, so to begin, the labs at the very bottom is very similar to lab 2 in which you've done. It's a workbook. And I want you to follow the same process. I've given you questions to answer, and I want you to do the labs on your own, but you don't submit those. But you have to do those labs to understand the course and to do well on the exams. Okay, so make sure you're doing those. All right. Now, to assist you, right off the bat, there are 2 videos. And the first video is on meiosis, the process of meiosis. So you can read it in the workbook, but many of you are better visual learners. And I think a lot of what you're going to get out of this brief introduction by America's favorite high school teacher, Mr. Anderson, on meiosis is actually very good. And, in a 10 to 12 minute sort of presentation. I think this is actually 9, so I got a really short one here for you. Now, because I really want the idea of independent assortment and what that means. Video number 2, called Meiosis and Mitosis with Beads, by Mr. Anderson, is going to sort of take you through how independent assortment in chromosomes actually works. But I will recommend this, that you want to get through the workbook, at least through the readings, right? Have a solid foundation on the readings, even before you do any of the lab exercises, before you look at the simulation. And then you can really understand, how independent assortment works. So my history in the past of teaching it like this is that this will catch 90% of you. You'll get it, right? And then it'll be a little abstract to you guys, who are the history majors and stuff, kind of like me, the anthropology guy, who really didn't get it the first time. That's when you should reach out to me, because I can work with you. We can work together on this, and sort of I can help you through this idea of independent assortment if you don't get it. Okay, but I think these 2 videos might help you, especially the second one. Now, when we talk about the heterozygous advantage, trying to figure out, something is a disease or not, and how we can sort of predict a disease, I've got a film called Malaria and Sickle Cell Anemia for you to look at. It also talks about the history of it, how it was discovered, how the genetics of it was discovered, and also trying to figure out, in what context, is malaria good or bad? So that film is very important for you to look at too, right? And then, of course, at the very bottom of the page is lab number 3 on inheritance. And this term, we're turning them in like all the other papers. We're just doing it over email. And that's simply because the assignment stuff, people are uploading them. I've heard tales of this because the system is so overloaded because everyone's going back to school. And by the way, we don't even keep any of the material here at DVC. This is like some centralized mainframe somewhere in Oshkosh, Wisconsin. So when you call the help desk, you're talking to somebody 2,000 miles away, and they're going to ask you what school you're from, and you realize, oh no, all my work is on some relational database server that may not be functioning correctly or not. So what happens is if you send the work directly to me, it bypasses all that, ends up in my Dropbox, in my mailbox, or even through the campus mail here, or a Canvas mail, and it's solidly in there. I know that I've got it from you. So that's why we're going to do it now. If I'm teaching this course again next year when things settle down and they build the infrastructure up, I may go back to regular submissions. But I just want to let you know why I'm doing it. All right, so let's get to work on that. Any questions? You know who to call. Right here. Bye.